Hello everyone, and welcome to this audio podcast today. Looking at as part of this series the cytometry cyclone. This audio podcast is part of the Rakas and Bombard episode, and this audio podcast as part of a Storm Robotics production. Today looking at and sort of continuing in and around T cell activation, allo recognition, and allo response in human renal transplantation. So on acute cellular acute cellular as part sort of part of the as part of the sort of intracellular signaling pathways in T cell activation. Acute cellular rejection is mediated by lymphocytes, which become alloactivated against donor antigens. In vivo stimulation of alloreactive T cells takes place primarily in the peripheral lymphoid tissue of recipients, although sensitization may occur. One of the sort of strongest stimulus in context of immunobiology is a stimulus provided by donor dendritic cells. Donor dendritic cells, also referred to as passenger leukocytes, in and as part of the allograft, and as part of the transplanted organ, which enter circulation and end on the lymphoid tissues of a recipient. These dendritic cells function alongside the antigen presenting cells, and provide a stimulus of hemoleukocyte antigen HLA class II reactive CD4 cells, which then stimulate growth and differentiation of HLA class 1 reactive CD8 positive cytolytic lymphocytes. CD8 positive cytolytic lymphocytes, also referred to as CD8 positive, or can be referred to in amongst the literature as CD8 positive cytotoxic T cells. And alloreactive lymphocytes enter circulation, and these react with the allograft vascular endothelium, a primary target for initial stage of prompting cellular rejection. The lymphocyte endothelial interactions depend on the expression of appropriate target antigens on the endothelium. HLA class 1 antigens are constitutively expressed at the cell surface area at the cell surface, whereas HLA class 2 antigens expression must be induced by various cytokines, especially gamma interferon. And furthermore, adhesion molecules such as intracellular adhesion molecule and vascular adhesion molecule, and vascular adhesion molecules, i.e. ICAM or VCAM, integrins and selectins, which also play a critical role amongst cell surface interactions between lymphocytes and endothelial cells and activation. So such molecules release of cytokines and inflammatory mediators. These processes then lead to a migration of lymphocytes through the vascular wall. That draws this audio podcast to a close. This audio podcast is part of the Cytometry Cyclone. This audio podcast is part of the Rakas and Bombard episode. And this audio podcast is part of a Storm Robotics production.